Hi, everyone. Welcome to Naturally Immune for Life. Thank you for joining me today, and thank you so much for your time. Today, we're going to continue with nutrition and the microbiome, specifically regarding pediatric health, but bear in mind and keep in mind that this can affect anyone at any time, which is malnutrition and poor gut health. And doesn't matter if you are a young adult, an adult, or an elderly person. Um, these types of symptoms and syndromes can affect anyone at any time, simply due to poor diet and poor gut health. So let's get started. And I'm going to be reading here. And this is a little long, so bear with me. This is derived from the source, Building Better Barriers, How Nutrition and Undernutrition Impact Pediatric Intestinal Health. And you'll see, so that you don't think I just cherry pick things and put them together on my own, everything I've been saying in many places on this website, including the About page, is all reiterated in this one paper, pretty much step by step and point by point and factor by factor. So let me get to the top here. Microbiome health shapes nutrition and immunity. Again, nutrition comes from the inside. It is not simply what you eat and then hope to derive benefit from. What you eat has to sustain the microbiome, and that in turn leads to a healthy outcome. Nutrition does more than sustain life. It directly shapes the immune system and the microbial landscape of the gut. Malnutrition, particularly in children, remains a devastating public health issue with long-term impacts on immunity and development. Emerging research reveals how early diet, immune function, and the gut microbiome are deeply intertwined, influencing lifelong health. So this, in part, contains both the initial development of the infant gut and how it transitions into the healthy, mature microbiota. Let's explore how nutritional status affects gut immunity, microbial development, what goes wrong during malnutrition, and how innovative dietary interventions may help reset this delicate balance. The intestine, the body's largest organ. The intestine is not only central to digestion, it's the largest immune organ in the body. It contains specialized clusters of immune tissue called gut-associated lymphoid tissue, GALT, commonly known as GALT. And it is densely populated by both innate and adaptive immune cells throughout its layers, from the epithelial surface to the submucosa, lamina propria, and even the muscle layers. These immune cells perform two essential and sometimes opposing tasks: tolerance, or the suppression of immune responses to harmless substances like food proteins or commensal microbes; activation, or the rapid defense against harmful pathogens. Nutrition is critical in regulating this balance. Nutrients such as vitamin D, retinoic acid, and short-chain fatty acids influence how these immune cells develop, migrate, and respond within the gut. How nutrition shapes the immune landscape. The development of GALT and its immune cell networks is highly dependent on how nutrients are delivered. Enteral feeding, where nutrients pass through the digestive tract, supports GALT development and mucosal immunity. Parenteral nutrition, or intravenous feeding, on the other hand, leads to GALT shrinkage and weakens gut immune function. Animal models show that fasting, even as brief as 12 hours, can cause measurable reductions in GALT size. Now, this isn't so much of a problem for those who aren't malnourished, but as you'll see in a minute, in severely undernourished children, post-mortem studies confirm this atrophy. So, of course, we're talking about children who died from malnutrition. Clinically, enterally nourished patients have stronger immune responses, fewer infections, and shorter hospital stays compared to those on IV nutrition or suffering from prolonged undernutrition. Secretory IgA, the frontline defense compromised by undernutrition. A key player in gut immune defense is secretory immunoglobulin A, SIgA. This antibody coats the intestinal lining, targeting microbes to prevent them from breaching the mucosal barrier. It acts both as a defender against pathogens and a curator of the microbiome by selectively binding beneficial bacteria. Undernourished children often have lower levels of SIgA or produce antibodies that fail to properly discriminate between helpful and harmful microbes. This breakdown contributes to barrier dysfunction and systemic inflammation, and has even been shown to cause weight loss when microbiota from undernourished children are transferred to animal models. One reason this happens is that nutrient deprivation alters the glycans on bacterial surfaces, disrupting SIgA's ability to recognize the good guys from the bad. And if you go back a couple of posts to why you need galactose, that will help shed further light on this function. SIgA is the most common immune deficiency syndrome. The microbiome, a key regulator of gut function and immunity. The intestinal microbiome plays an integral role in gut health. It contributes to immune development, primes immune responses, and strengthens the intestinal epithelial barrier. And how it does this is commensal microbes, beneficial bacteria, compete with pathogens for space and nutrients, reducing infection risk and influencing host metabolism. Early colonizers like Bifido, Clostridium, and Bacteroids interact directly with the gut mucosa, helping to shape immune tolerance and tissue development. Lactobacillus and Akkermansia, not quite sure how to say that, stimulate mucus production and reinforce the epithelial barrier, key defenses against microbial translocation. Intestinal epithelial cells sense bacteria and their byproducts through pattern recognition receptors, like toll-like receptors. These interactions regulate tight junction integrity, mucus and antimicrobial peptide production, and epithelial cell renewal. This symbiotic relationship is essential to gut homeostasis and highly sensitive to diet. Microbes help digest nutrients that infants can't. In infants, certain gut bacteria provide critical enzymatic support that compensates for immature digestion. These microbes break down human milk oleosaccharides, HMOs, providing calories and immune modulating molecules that infants cannot extract on their own. They produce short chain fatty acids like acetate, propionate, and butyrate, beneficial fermentation products that help seal the gut barrier and modulate inflammation. They also synthesize essential vitamins such as B12 and folate. For example, term infants with microbiomes dominated by bifido and bacteroids produce more short-chain fatty acids than low birth weight infants. These bacteria's genomes encode enzymes that metabolize over 200 unique milk oleosaccharides, and these are HMOs, demonstrating how dependent infant nutrition is on a well-developed microbiome. Sorry, it's cool and rainy again. I don't want to lose my voice. How malnutrition disrupts microbial harmony. Undernutrition alters the composition and function of the gut microbiota in significant ways. Microbial diversity is reduced, commensal species decline, while facilitative pathogens like E. coli and staph proliferate. Oxidative stress increases due to the loss of dietary antioxidants like vitamin C and E, changing gut redox balance and favoring inflammatory microbes. These microbial changes have profound effects with decreased short-chain fatty acid production, weakening the epithelial tight junctions, and persistent immune activation even after calorie intake is restored. In short, the malnourished microbiome becomes a self-reinforcing source of inflammation, worsening the very symptoms it helped create. Rebuilding the gut is more than calories. Providing food isn't enough to reverse the effects of undernutrition. Restoring a healthy microbiome and immune function requires targeted nutritional therapies. A striking example comes from Bangladesh, where children with moderate acute malnutrition, so we're not even talking about severe acute malnutrition here, were given a microbiota-directed complementary food, MDCF2, compared to these receiving standard ready-to-use supplement, RUSF, or sometimes known as RUTF. The MDCF2 group showed better weight and length gains, improved plasma markers related to brain and bone development, restoration of key microbial species and their metabolites. These results suggest that microbiota-targeted diets could provide long-term benefits, promoting recovery that goes beyond weight gain to address immune competence and cognitive outcomes. Maintaining this sustained shift to a mature and diverse gut microbiome is critical to preventing long-term negative consequences of undernutrition, such as neurodevelopmental delays and the development of obesity and metabolic disease later in life. Modulating the intestinal microbiome in undernutrition has the potential to improve intestinal barrier and immune function, as well as host energy extraction to improve long-term outcomes. The challenge of translating animal research. Much of our knowledge comes from rodent models, which have important limitations. Rodents are born with immature intestines, unlike humans, whose neonatal guts are more developed. Murine GALT forms early and is B-cell dominant, while human GALT forms postnatally and is more T-cell rich. Dietary responses differ, making it hard to replicate human malnutrition accurately. Pigs and guinea pigs offer better analogs due to their similar gut development, but cost and complexity limit their use. Non-human primates are more comparable, but also expensive and ethically complex. A new frontier, human enteroid models. A promising alternative is the use of human enteroids, lab-grown stem cell-derived miniature intestines. These models mimic real human intestinal tissue, including immune cell interactions. They can be derived from healthy or malnourished donors for personalized research. They allow for testing of microbial, dietary, and pharmaceutical interventions in a controlled setting. Already used to study diseases like necrotizing enterocolitis and viral infections, enteroids could become key tools in understanding undernutrition and guiding future interventions. And when it comes to necrotizing enterocolitis, I'm guessing you might or might not know that can be caused by infant formula. So I'm guessing this might be one of the reasons they are switching to this enteroid model, because you cannot ethically use infants or preterm infants for guinea pigs. So what we still don't know, despite tremendous progress, critical gaps remain. Which nutrients or metabolites are essential for rebuilding intestinal immunity? How do different types of undernutrition, that is protein versus micronutrient deficiencies, alter gut immune signaling? What are specific microbial species or functions required for recovery? Can we define a healthy microbiome universally, or must it be population specific? What maternal, prenatal, or feeding practices shape microbiome development most effectively? Answering these questions will require collaboration between nutritionists, microbiologists, immunologists, and public health experts, along with better models and long-term clinical data. Conclusions: a systems biology approach to nutrition. Undernutrition is not simply a problem of insufficient calories. It is a systems level disorder that disrupts the immune system, damages the intestinal barrier, and derails microbiome development. Effective solutions must go beyond feeding programs to address the complex biological feedback loops involved. Nutrition, when designed with the microbiome and immune system in mind, becomes a powerful tool not only to save lives, but to build stronger and more resilient bodies and minds. Now, this is going to take years to get to where the Japanese already are, GCMAF and other MAF products, which addresses several key things in that list. One, they're using universal microbes and universal donors, which would be bovine colostrum, which is considered a universal donor. And they are understanding that the substrate necessary to affect the immune system is lactose and galactose. So a lot of this, and the Japanese, especially when something is non-harmful and beneficial, it's already on the shelf. So they don't wait. If something is safe and harmless, they do not wait to take the years and years and years for these studies and clinical trials to be completed. In this 2 steps alone, one choosing nutrient-dense foods that are complementary and help form the maturation of the microbiome, and adding a specific probiotic to these nutritional foods, nutrient-dense foods, which also changed outcomes. These alone have taken probably at least 15 years. So what are these nutrient-dense nutritional food ingredients for acute malnutrition? In case you were ever curious, this specific recipes, the ingredients that they used in these types of studies, and they used for therapeutic foods to combat malnutrition are chickpea flour, soy flour, peanut flour, banana powder, oil, often soybean or canola, sugar, a small amount, and a vitamin and mineral premix, which includes iron, zinc, calcium, vitamins A, D, E, B complex, etc. These ingredients were selected based on their ability to promote beneficial gut bacteria, enhance microbial diversity, and improve host metabolism and growth signaling. So this was enacting some specific changes to the ready-to-use supplementary food, which the ingredients are usually peanut paste, vegetable oil, soy or canola, sugar, dried skimmed milk or whey protein, maltodextrin or cereal flour, usually rice or corn, and the vitamin mineral premix. RUSF is highly effective for weight gain and nutrient delivery, but it does not specifically target microbiota restoration. So, in addition to this particular research, the work of Dr. Jeffrey Gordon and team created and tested the addition of a single probiotic to RUTF to improve outcomes. So gut harmony, the essential role of B12, folate, and short-chain fatty acids in digestive and whole body health. Vitamin B12, folate, and short-chain fatty acids play essential roles in maintaining overall health, especially gut and brain function. Vitamin B12 is crucial for red blood cell formation, DNA synthesis, and neurological function. In the gut, it acts as a natural antidote to hydrogen sulfide, a toxic gas produced by some bacteria during fermentation. In conditions like small intestinal bacterial overgrowth, excess H2S can damage the intestinal lining, disrupt energy production, and worsen symptoms such as bloating and fatigue. It's also a gasotransmitter, and it's rocket fuel for cancer, and SIBO also increases malabsorption, especially H2S SIBO. B12 helps neutralize this compound and supports healthy cellular metabolism. Folate, vitamin B9, another key B vitamin, works alongside B12. B12 and methylation pathways that regulate gene expression, detoxification, and neurotransmitter production. Folate deficiency can lead to anemia, immune dysfunction, and impaired gut barrier repair. Short-chain fatty acids such as acetate, propionate, and butyrate are beneficial byproducts of fiber fermentation by gut bacteria. They serve as fuel for intestinal cells, reduce inflammation, support tight junction integrity, and modulate appetite and insulin sensitivity. Butyrate, in particular, helps maintain the gut-brain axis and protects against colon cancer. Together, B12, folate, and short-chain fatty acids support a healthy microbiome, immune tolerance, and optimal energy metabolism, key pillars of long-term health. So, one last little snippet here. How probiotics boost B vitamin production for better health. Certain probiotics play a crucial role in enhancing the production and availability of B vitamins within the gut, supporting overall health. Species from the genera Lactobacillus and Bifidobacterium are especially known for their ability to synthesize essential B vitamins like B12, folate, and riboflavin. These beneficial bacteria produce B vitamins as metabolic byproducts during the fermentation of dietary fibers and other substrates, which can then be absorbed by the host. For example, Lactobacillus reuteri and Bifidobacterium longum have been documented to contribute significantly to folate production in the intestine. Additionally, these probiotics may enhance the gut environment, promoting the growth of other beneficial microbes that contribute to vitamin synthesis. By increasing B vitamin availability, probiotics support critical functions such as DNA synthesis, red blood cell formation, and nervous system health. This symbiotic relationship between gut microbes and the host highlights the importance of maintaining a healthy microbiome for optimal nutrient status. So what can you do? In addition to natural oral immune therapy like MAF or GCMAF, or of course, my preferred route, which will eventually flush out bad bacteria, and that will happen in 4 to 6 months. And it will also help reduce toxins. You can use beneficial yeasts to do all this faster with beneficial yeasts like Florastor. So if there is something already clinically studied to help eliminate these very specific bacteria that are already known to be affecting children like this, and producing chronic diarrhea, as well as hospital-acquired infections, and et cetera, et cetera, et cetera. Hospital-acquired gut dysfunction in general, from whether it's chemo or overuse of antibiotics or what have you. Again, this helps promote bacteria you don't want while decreasing bacteria you do want. So beneficial yeasts like Florastor can help speed that up a little bit as you are restoring beneficial bacteria and giving yourself all of the substrates, the beneficial yeast, the beneficial probiotics, and the colostrum that are all universal and safe. If they're in the grocery store, you can pretty much, or actually being sold as a probiotic, you can pretty much be assured that these are universal and generally not harmful to anyone. So that's something you can just do, or anytime you feel like you've had a bad diet for too long, anytime you feel like you're malnourished, you can do a round or two of florist store and then get right back on the program of natural oral immune therapy as you bring yourself back to optimum gut health and optimum nutrition. So I just would like to say that I wasn't really planning on doing this for this site, but I truly would like a favor. If you would click on my affiliate links and order a product in the very near future, because I've been informed by Amazon that my account is already at risk because I have taken far too much time in rebuilding these sites and transferring these sites and getting everything up and running, that I should have waited to re-sign up to be an Amazon affiliate, and then I could have begun reviewing and referring and listing specific products that I use and like. And for the most part on this site, they're all going to be food-based supplements because those are the only things I actually use. And but I do often run across some very good probiotics, and I do run across some less expensive alternatives to my favorite colostrum, and I do use them. One happens to be out of stock at the moment, so it's not listed, but these are the types of things I send along with my daughter when she leaves for school, and I also give them to my animals, and I also use them because they're very good. And sometimes I just toss a few extras in there so I don't have to use my more expensive Body Boost, which is my favorite brand of bovine colostrum. So I would truly, truly, truly appreciate that. And I'll be adding more as time goes on. But essentially, they're going to be food-based supplements. And the only two that I can actually think of that aren't food-based would be slippery elm and marshmallow root. And those are two herbal products that just help seal the gut and highly reduce inflammation as you are transitioning to natural oral immune therapy. So those would be the only two that I can actually think of, aside from a beauty product that maybe castor oil. Those are the only ones I could think of that I would actually be promoting. And I'm going to watch myself on this site so that I don't end up with another scarlet letter. So I really wasn't planning on doing that. But since it appears the Amazon Associates has changed their conditions and rules from the last time I joined way back in 2004, I guess that was why I desperately need this favor. So I thank you for doing that for me, my friends, and I'll see you next time. You have a great day and a great week. Bye.